Okay, now that we know that the cell membrane is very fluid and it looks like a mosaic, now we would like to talk about what are the factors that affect the fluidity of these cell membranes. So not all cell membranes are the same in terms of fluidity, and there's two main determinants that determines whether or not a cell membrane is fluid. The first is the fatty acid content. As you recall, we have those bent legs that are made up of the unsaturated fatty acids. So the more unsaturated fatty acids you would have, then the less compact a cell membrane is, meaning the less rigid a cell membrane is. Meanwhile, if you have more of these viscous and straight shaped fatty acids, as you can see in the case of saturated fatty acids, then you would have this very compact form, and that exactly is what will make a membrane more less fluid and more rigid. Another factor that determines the fluidity of a cell membrane is the cholesterol content. So the more cholesterol you have in between these rigid saturated fatty acids, then you would have a less rigidity and more fluidity. So those are the two factors that determine the fluidity of a cell membrane. Firstly is this fatty acid content, is it more saturated or less saturated? And then the second is the cholesterol content. Now, this fluid mosaic model that we talked about is also what contributes to the permeability of the cell membranes. So there's a lot of three different terms that you need to be familiar with when talking about cell membrane permeability. There's a term called impermeable, permeable, and semi-permeable. Impermeable means that no compounds can enter through the membrane. Permeable means that all compounds can enter through the membrane, meaning both solvents and solutes, while semi-permeable membranes really usually indicates that membranes in which certain compounds can enter through, and this is usually solvents only. Now, this brings us to transportation across cell membranes. So when we talk about how a solute or a solvent is transported into a cell membrane or outside of a cell membrane, then there's two different types of transportation, whether or not we are using a protein helper or not. So if you need help from a transporter protein, then that transportation is facilitated. If you don't need help with a transporter protein, then it's a free transport, meaning that solutes move across freely without the use of a transport protein. Now, these facilitated transports can be facilitated by channel proteins or transportants. So what's the difference between a channel protein and a transporter is that channel proteins they contain polar pores, whilst transporter proteins are those that can actually recognize the molecule and then will change conformation. Basically, when we talk about a channel, you're looking at a cylinder shape structure in which it doesn't really change conformation. It just sort of lets the solute through. Meanwhile, with transporters, you would expect that they would actually change shape when they are doing the transportation. Now, these are two examples of transporters. I won't go into detail exactly on how they work because we'll be talking about these in the next systems block, but basically the examples of transporters includes the aquaporin transporter of water in the renal tubules, and also the glucose transporter, which is found in almost all cells and regulates the influx of glucose into the cell. Now, this is an example of a channel, and this is what we find mostly in signal transduction, particularly in the nervous system. So we have voltage-gated sodium channels which can open and shut depending on the membrane potential of a cell. So these are just examples, and we don't need to talk about them in detail at this present block. Now, but as we talked about just now, is how cell membrane transport classified based on the proteins they use. Now, they can also be classified in terms of whether or not they need energy to do the transport. So there's such thing as passive transport and active transport. When we talk about passive transport, it means that the cell membrane does not need any energy for the transportation of certain solutes. Well, when we talk about active transport, it means that we will need ATP or energy. So there's some different kinds of passive transport, such as simple diffusion, facilitated diffusion, or osmosis. And this indicates diffusions of several solutes, and like carbon dioxide or oxygen, glucose and water. And they move from a place or an area with more concentrations of that solute to an area with less. And because of this, then they don't really require energy, because it's a passive movement. Meanwhile, when we're talking about active transport, a lot of it occurs because we need to move molecules from a low concentration to a high concentration gradient, and this does require a lot of energy because you're sort of going against the flow of nature. So let's try and illustrate them one by one. So here we'll look at pay attention to the first transport of a molecule. This is basically a simple diffusion, where we see a large concentration of this molecule moving to a low concentrated area. Because of its this natural tendency to flow from an area of high concentration to an area of lower concentration, then we don't really need energy for this, and this is called a passive transport. Specifically, it's called simple diffusion because we don't really need a transporter for this. But if, for example, in order to move through into this less concentrated gradient, we will need the help of a channel like this, then we call this a facilitated passive transport using a channel. And for example, please mind my poor animation skills, but basically this is, if for example you need this protein that actually changes shape or conformation upon the transportation process, then we call this a facilitated diffusion by carrier protein. So in this protein helper, we don't expect to see any change in shape or conformation. That's why we call it a channel. And in this structure, we see a change in conformation, and that we really call a facilitated diffusion using a transmembrane carrier protein. And this is basically the movement of water from a higher water volume to a lower area of water volume, and this is, as you know, is called osmosis. And these are all a form of passive transport. Meanwhile, in the case of active transport, the key concept here is that it requires energy. So in this case, we have this funny looking transporter which changes shape upon conformation and also requires energy. And for this, we call this particular process active transport. Alright, so now we can also, moving on to another classification process of transportation based on the amount of molecules. I think we just skim this one. Basically, if you're transporting only one molecule at a time, like this glucose going into the next space, then we call this a uniport. Meanwhile, if you're transporting two molecules at the same time in the same direction, then you would call that a symport. And if you're transporting two molecules at the same time in opposite direction, as you can see in number 3, then we would call that an antiport. So this is just based on the amount of molecules that is transported at the time.